You think you know what lives on your skin? Maybe you washed your face this morning, scrubbed behind your ears, thought you were clean. But right now, as you listen to this, millions of creatures are crawling across your eyelashes, burrowing into your pores, feeding on your dead skin, mating on your forehead. They are with you when you wake. They are with you when you sleep. They have been living on humans for over 3 million years, and you never invited them in. 8 legs, translucent bodies, mouths designed to consume you piece by piece. What are they? Why do they choose your face? And what happens when their numbers explode? Small, you need magnification to see them. Demodex follicularum, Demodex brevis, Sarcoptes scabiei, Dermatophagoides terranicinus, Trombiculidae larvae. These are not passengers. They are permanent residents. Some live in harmony with you. Others burrow tunnels through your epidermis and lay eggs in your flesh. The mites on your body outnumber the hairs on your head. According to research published in Molecular Biology and Evolution in June 2022, prevalence in adult humans likely exceeds 90%. A study from the University of West Attica published in Parasitologia in April 2024 found 66% of examined subjects tested positive for demodicosis at densities above 5 mites per square centimeter. That is not an infestation. That is baseline. If you value documented parasitology and dermatological history like this, subscribe. It is the best way to support our work. Also comment from which city you are hearing this from. This script is researched and factual using sources from peer-reviewed journals including Clinical Ophthalmology, Volume 18, published June 2024 by Kiovianu and team at the University of Medicine and Pharmacy, Carol Davila, and the Global. Prevalence meta-analysis published in Science Direct June 11, 2025 by Maliki and colleagues at Shahid Beheshti University, covering data from January 2000 to January 2025 across multiple WHO regions. Now, back to the information. Lives in the hair follicle above the sebaceous gland. Feeds on sebum and dead skin cells. The mite has 8 legs in its adult form, a segmented body covered in scales and sharp mouthparts designed to pierce follicle walls. Research from North Carolina State University published in PLOS 1 in August 2014 using molecular detection methods found 100% of adults over age 18 harbor at least one Demodex species. 100%, not most people, everyone. Each human being carries between 1,000 and 2,000 mites according to studies cited in the journal published by the National Center for Biotechnology Information in July 2017. At night, they emerge. Researchers estimate more than 1.5 million mites on an average human body come out to mate and consume built-up sebum around hair shafts, as reported by PBS News. And USDA entomologists in September 2016. They move at 8 to 16 centimeters per hour across your skin surface. The female deposits eggs inside the follicle. Each egg is oval, 0.1 millimeters long. Incubation takes 3 to 4 days. Larvae have 6 legs. Nymphs develop 8. The full life cycle spans 10 to 17 days on the host. Without a host, they survive only 72 hours. The mites are most dense near the nose, on the forehead, around the eyebrows, and along the eyelashes. In a study of 184 individuals published in Greece in April 2024, 70% of patients were female and 30% male, but 64% of demodicosis positive individuals were female. The density peaks between ages 20 and 30 when sebum production is highest, then plateaus. By age 60, the prevalence reaches 59% according to the meta-analysis published June 2025. The mites do not bite. They do not suck blood. They chew. Their saliva contains digestive enzymes that break down skin cell walls. They eat the liquefied tissue. Most people never feel them. Your immune system identifies the mites as part of your body, not invaders. This is commensalism at its most intimate. But density matters. Above 5 mites per square centimeter, symptoms begin. Inflammatory acne lesions, papules, pustules, eyelash crusting. The condition is called demodicosis. Primary demodicosis occurs when mite overpopulation alone causes symptoms. Secondary demodicosis happens when other skin diseases weaken immune response and mites multiply unchecked. Rosacea patients show significantly higher Demodex densities than healthy controls, as documented in multiple dermatology journals. Patients with end-stage renal failure, diabetes, Behçet disease, and those taking immunosuppressive drugs all show elevated mite counts. A Thai study published in Frontiers in Public Health in January 2025 found that most Thai people possess limited knowledge about Demodex, despite their universal presence. The mites can introduce bacteria into the dermis during feeding. Staphylococcus has been found in secondary demodicosis cases. Treatment involves topical or oral antiparasitic medications. permethrin cream, ivermectin. But the mites return. They always return, because they live on every human you touch. Between sexual partners. A study published in September 2024 in Experimental and Applied Acarology found two distinct morphotypes of Demodex follicularum, short and long forms, segregated by microhabitat on skin versus eyelashes. Both forms show statistically supported double infections where individuals host both populations simultaneously. The mites cannot jump, they cannot fly, they crawl from face to face during close contact. Shared makeup brushes, shared towels, shared pillows. Each surface becomes a vector. Once separated from skin, the mites can survive on fabrics for hours to days before finding a new host. You inherited your mites. Research published in the Proceedings of the National Academy of Sciences, analyzing 70 human hosts from diverse ancestries, found 4 distinct genetic clades of Demodex. Follicularum distributed by geographic origin. People of African descent carry all 4 clades. People of European descent carry predominantly one clade. The last common ancestor of these 4 groups lived over 3 million years ago, meaning the mites predate modern humans and the two species evolved together. As humans left Africa and spread across continents, certain mite lineages were lost. The mites on your face tell the story of your ancestors. Genetic analysis shows Demodex brevis has greater diversity than follicularum. Within single individuals, multiple lineages of brevis coexist. A study from North Carolina State found brevis samples from one face exhibited more genetic variation than follicularum samples from Chinese, North American, and South American populations combined. Chinese brevis populations form a separate clade from American populations with deep genetic divergence, suggesting strong geographic isolation. The distance between these clades is comparable to differences between subspecies and other organisms. The mite feeds on sebum and gland cell contents. Unlike follicularum, which clusters in groups of 3 to 6 individuals in the follicle infundibulum, brevis lives solitarily. Each gland houses at most a few individuals. The life cycle mirrors follicularum. Egg to larva to nymph to adult. 17 days, females lay eggs inside the gland. The young develop hidden from view. Only molecular detection methods reveal their presence because standard skin surface biopsies often miss them buried in gland tissue. A review in Clinical Ophthalmology published June 2024 noted that ocular demodicosis incidence ranges from 29% to 91% across geographic locations and study populations. Brevis is detected more frequently in ophthalmology patients, who showed a 57% prevalence in the global meta-analysis. Chronic severe rosacea and brevis infestation often coexist, making treatment challenging. The mites may act as vectors for bacteria. Their bodies and waste products trigger inflammatory cascades. Diagnosis requires either skin biopsy with histological examination or molecular PCR testing. Standard surface sampling misses brevis because the mites hide deep in glands. Cellophane tape methods show a 91% positive rate for follicularum, but only 34% for brevis, as documented in standardized studies. Treatment mirrors. Follicularum protocols, but success rates vary. Topical tea tree oil, oral ivermectin at specific dosages, intense pulsed light therapy to reduce gland inflammation. Some patients undergo lid scrubs with diluted baby shampoo to mechanically remove mite debris from eyelid margins. But brevis resists eradication. The glands provide shelter. The mites reproduce faster than treatment can eliminate them. And reinfection from untreated contacts occurs within weeks. The immune response determines outcome. Immunocompetent individuals tolerate moderate brevis populations without symptoms. Immunocompromised patients develop crusted lesions and massive infestations. The mites win when the host weakens. 0.2 to 0.24 millimeters for males. This mite does not live on the surface. It burrows. Sarcoptes scabiei was first identified by Italian biologists Giovanni Cosimo Bonomo and Giacinto Sistoni in 1687, making scabies the first human disease with a known microscopic causative agent, as noted in historical records. The mite belongs to family Sarcoptidae, order Astigmata, class Arachnida. The body is oval. The dorsum has transverse ridges and triangular scales. 4 pairs of legs. The first 2 pairs have long unsegmented pedicels with caruncles, sucker-like structures for gripping skin. Males have these on the 4th pair too. The mouthparts are chelicerae designed for cutting. The tunnel appears as a raised serpentine line, grayish or skin-colored, 1 centimeter or more long. Inside this burrow, she deposits 2 to 3 eggs per day for about 2 months. Each egg is 0.1 to 0.15 millimeters long. Hatching occurs in 3 to 4 days. The larva has 6 legs. It molts to an 8-legged nymph. 2 more molts follow. Adult in 10 to 14 days. Total life cycle, 10 to 17 days. A female lays up to 30 eggs before dying at the end of her burrow. The mite feeds by ingesting and consuming intracellular fluid from broken cells that fill the burrow. Burrowing uses both cutting mouthparts and hooks on the legs. The process damages the epidermis. Scabies occurs worldwide, affecting all races and socioeconomic classes. As of 2015, it affects approximately 204 million people globally, 8% of the world population according to WHO data. The young and old are most commonly affected. Transmission requires 10 minutes of direct skin-to-skin contact for classic scabies. That is enough time for impregnated females to transfer between hosts. Transmission also occurs via fomites, bedding, clothing, towels, anywhere the mite survives off the host. Off human skin, Sarcoptes scabiei survives a maximum of 72 hours at room temperature. When a person is infested for the first time, symptoms do not appear for up to 2 months. The immune system needs time to recognize the mite antigens and mount a response. During this asymptomatic period, the person is contagious. If previously infested, symptoms appear within 1 to 4 days because the immune system is primed. The burrows are most frequently found between fingers, on wrists, elbows, knees, the penis, breasts, and shoulder. Blades. A papular rash appears on the buttocks, back, and abdomen even where mites are absent. This is sensitization from the immune response. Up to 2 million mites on a single individual according to StatPearl's data from July 2023. 2 million. The skin develops thick, hyperkeratotic plaques and crusts covering large areas. The palms, the soles, under fingernails. Itching is minimal because the immune system is too weak to respond. But contagion is extreme. A single crusted scabies patient can spark an outbreak. Hospital workers, nursing home residents. Entire wards have been infected from one index case. The crusts protect the mites from topical treatments, requiring prolonged therapy. Secondary bacterial infections are common because scratching breaks the skin. The mite, eggs, or fecal pellets confirm diagnosis, but sensitivity is low. Multiple scrapings from different sites increase detection. Treatment is permethrin 5% cream applied from neck down for 8 to 14 hours, then washed off. Repeat in 7 days. Oral ivermectin is an alternative. 2 doses, 2 weeks apart. All household contacts and sexual partners must be treated simultaneously, even if asymptomatic. Bedding and clothing used in the last 3 days must be washed in hot water above 60 degrees Celsius and dried in a hot dryer. Items that cannot be washed should be sealed in plastic bags for 72 hours. The mites starve. Symptoms can persist 2 to 4 weeks post-treatment because dead mite antigens remain in skin. If itching continues beyond 4 weeks, retreatment is needed. Scabies is a neglected tropical disease. WHO declared it so in 2009. Outbreaks still occur in refugee camps, prisons, and overcrowded housing. The mite respects no borders. The average human sheds 1.5 grams of skin per day. That is enough to feed millions of mites. Dermatophagoides, Tyrannus is the European house dust mite, first described by Vorhorst and colleagues in 1967 as the causative allergen for house dust allergy. Dermatophagoides farini is the American house dust mite. Both species measure 0.2 to 0.3 millimeters in length. Translucent bodies. Striated cuticle, 8 legs. The mites are cosmopolitan, found on every continent except Antarctica. In Australia, Tyrannus dominates in coastal areas. In drier regions, Farini prevails. In tropical and subtropical zones like Southeast Asia, Blomia tropicalis, a storage mite, coexists with both dermatophagoid species. Anywhere with humidity above 50% and temperature between 20 and 25 degrees Celsius. They cannot survive below 50% humidity for extended periods. Well-ventilated homes in dry climates contain few dust mites. Homes with sustained humidity above 50% can harbor over 100 dust mites per gram of dust. The life cycle spans 2 to 3 months, egg to larva to 2 nymph stages to adult. Females lay up to 100 eggs during their lifespan. Adults live 60 to 90 days. They feed by consuming skin scales and mold. Dermatophagoids, Farina shows preference for Alternaria alternata, Cladosporium sphaerospermum, and Wallemia sebifungi in laboratory studies. They avoid penicillium and aspergillus. Each mite produces 20 fecal pellets per day, and the pellets are 10 to 40 micrometers in diameter, similar to pollen grains. Small enough to become airborne, small enough to be inhaled. The fecal pellets contain digestive enzymes, peptidase 1, a cysteine protease. This enzyme persists in dried feces and becomes the major allergen trigger. The main allergens are DIRP1 and DIRP2 from Tyrannus, DIRF1 and DIRF2 from Farini. DIRP1 is responsible for triggering IgE antibody binding in 80 to 90% of dust mite allergic individuals, according to immunology studies. These are glycoproteins with proteolytic activity. When inhaled, they breach epithelial tight junctions in the airways. They stimulate protease-activated receptors, causing epithelial dysfunction and cytokine release. The immune system overreacts. Allergic rhinitis, asthma, atopic dermatitis. Up to 2/3 of asthmatic children and half of asthmatic adults have dust mite allergies, as documented by WHO. The allergens trigger atopic sensitization early in life. Infants exposed to high dust mite levels in the first year show increased risk of developing asthma by age 7. The relationship is dose-dependent. Higher mite densities correlate with worse symptoms. In Hong Kong, over 80% of asthmatic children are sensitized to house dust mites. In the Asia-Pacific region, dust mites are the major allergen in allergic rhinitis cases. Wash bedding weekly in water above 60 degrees Celsius. Hot tumble dry for 30 minutes after drying kills mites but not the allergen. Remove carpets. Use hard flooring. Avoid upholstered furniture. Reduce indoor humidity below 50% with dehumidifiers. Vacuum weekly with HEPA filters. But complete eradication is impossible. Even after aggressive cleaning, dead mite bodies and feces remain in dust for months. The allergens persist. Immunotherapy involves subcutaneous or sublingual administration of mite extracts to desensitize the immune system. Studies show immunotherapy reduces symptoms and medication use in dust mite allergic patients, but compliance requires years of regular treatment. This work draws from peer-reviewed sources including the dust mite meta-analysis and scientific reports published July 12, 2024 by Shuai and team at Capital Medical University Beijing using multiplex LAMP assays, and the Comprehensive Allergy Review in Asia Pacific Allergy, October 2014 by Yu and Tsai from Changgung University, covering environment evaluation and disease prevention strategies, validated in clinical trials. Now back to the story. Over 3,000 species exist worldwide, but only about 15 frequently bite humans. In the southeastern United States, the primary species is Utrombicula alfredugesi, also called Trombicula alfredugesi or Trombicula irritans. In Europe, Neotrombicula autumnalis dominates, called harvest mites, because they peak in late summer and autumn. In Asia Pacific, the genus Leptotrombidium includes species like Leptotrombidium deliens. These Asian species are medically important. They transmit Orientia tsutsugamushi, the bacterium causing scrub typhus. When a host brushes past, the larvae grab on. Clothing becomes a ladder. They migrate upward until they find skin or a barrier. Barriers like belts, elastic waistbands, sock tops, and collar seams cause clustering. The larvae cannot penetrate clothing but accumulate where fabric presses tight against skin. The larva pierces the epidermis with sharp mouth parts and injects saliva containing digestive enzymes. These enzymes liquefy skin cells, though hardened skin cells around the puncture form a feeding tube, the stylostome. The larva feeds through this tube for 3 to 5 days ingesting dissolved tissue. Then it drops off. The itching does not start during feeding. It begins 24 to 48 hours after the bite when the immune system reacts to the saliva left behind. The reaction is intense. Red welts 1 to 2 millimeters in diameter. Clustered bites around tight clothing areas, ankles, waistlines, behind the knees, groin, armpits. The welts persist for 2 to 3 weeks. Scratching breaks the skin and secondary bacterial infections follow. Geographic and seasonal distribution follows humidity and temperature. In the northern hemisphere, chigger larvae mature between June and September. Most human cases occur in summer and fall. In tropical regions, exposure happens year-round. Habitats include overgrown fields, wooded areas, ground near water, grassy lawns, golf courses, and parks. The mites are most numerous in early summer when vegetation is thickest. Occupational exposure affects harvest workers, hikers, gardeners, and military personnel training in endemic areas. Urban and suburban environments are not exempt. Overgrown lawns and gardens harbor populations. Larval mites acquire the infection before they ever feed. When they bite humans, the bacteria enter through the feeding site. Scrub typhus was historically confined to the Tsutsugamushi Triangle covering Asia Pacific and northern Australia. Recent reports document cases in Africa, southern Chile, and the Middle East. A study in North Carolina published in 2010 found evidence of rickettsia infections in U.S. chiggers, expanding the Osage endemic area. Scrub typhus symptoms include fever, headache, muscle pain, eschar at the bite site, and in severe cases, multi-organ failure. Mortality without treatment ranges from 0 to 70% depending on bacterial strain. Treatment is doxycycline. Shower within 1 hour of exposure to wash off larvae before they attach. Once attached, larvae fall off easily with scratching or washing. Nail polish and petroleum jelly do not work because larvae do not burrow. Home remedies suffocate nothing. The stylostome remains even after the larva detaches, continuing to cause itching for weeks. Treatment is symptomatic. Topical corticosteroids, oral antihistamines, calamine lotion, cold compresses. If secondary infection occurs, antibiotics are needed. The key is avoidance. Stay out of tall grass during peak season. Mow lawns, clear brush, remove leaf litter, reduce habitat. The larvae cannot fly, they cannot jump. They wait on vegetation. If you do not go to them, they cannot reach you. Sarcoptes burrows and reproduces in skin tunnels, triggering immune chaos. Dermatophagoids fills your home and turns your shed skin into airborne allergens. Trombiculidae larvae inject enzymes and create feeding tubes in your epidermis. They share one trait. They are arthropods. Class Arachnida, 8 legs in adults, 6 in some larvae. They evolved over millions of years to exploit mammalian hosts. They adapted to human expansion, following us into every climate and continent. They cannot be eradicated. They can only be managed. 65 to 130 million with dust mite allergies, thousands infected with scrub typhus annually. These are not rare events. These are baseline human conditions. The mites are not invaders, they are residents. Some became residents 3 million years ago. Others jumped species within the last few thousand years as humans domesticated animals and built permanent settlements. Dust mites evolved from nest mites that fed on bird skin. They adapted to human homes when we created stable indoor environments with mattresses and carpets. Demodex co-evolved with primates, diversifying as human populations spread and isolated. Sarcoptes crossed from other mammals, specializing into the human-specific variety homines. Each mite tells a story of adaptation and exploitation. Healthy individuals tolerate low mite densities without symptoms. The mites are commensal or symbiotic at normal levels, but immunity falters with age, disease, stress, or immunosuppressive treatment. Then the mites multiply. Demodex densities explode, causing rosacea and blepharitis. Scabies spreads unchecked in nursing homes and hospitals. Dust mite allergens trigger asthma attacks. The balance tips. Host and parasite enter conflict. Treatment can suppress populations, but not eliminate them. Permethrin kills scabies mites, but reinfection occurs if contacts are not treated. Ivermectin reduces Demodex counts, but the mites recolonize within weeks. Dust mite allergen avoidance lowers exposure, but mites persist in every home. Chigger bites resolve when larvae detach, but next season brings new larvae. Molecular PCR detects mites at lower densities than microscopy. Immunotherapy for dust mite allergies shows promise in clinical trials. New antiparasitic compounds target mite reproduction. Genome sequencing of Demodex folliculorum, published in 2022, revealed the smallest genome among panarthropods, 9,707 genes. This extreme genome reduction suggests the mites are transitioning from ectoparasites to obligate symbionts. They have lost genes for UV protection, melatonin synthesis, and many other functions because they live entirely within or on the host. They cannot survive without humans. We are their world. Understanding mite biology leads to better treatments. Identifying mite allergen epitopes enables component-resolved diagnosis, distinguishing true dust mite allergy from cross-reactivity with other allergens, like shellfish tropomyosin. Tracking Demodex genetic diversity reveals human migration patterns because the mites preserve ancient lineages lost in human populations. Demodex are crawling along your eyelash roots. They are feeding inside your facial pores. At night, more will emerge. If you sit on upholstered furniture, dust mites in the fabric release fecal pellets you will inhale. If you walk through tall grass next summer, chigger larvae will climb your shoes and find your ankles. If you shake hands with someone carrying scabies and hold contact for 10 minutes, mites will transfer. These are not possibilities, these are certainties. The mites have been here longer than you. They will be here when you are gone. They are not separate from humanity. They are part of the human condition. The lead researcher, Michael Palapoli, described collecting 70 human samples from diverse ancestries to map mite genetic diversity. 4 clades emerged. African populations hosted all 4. European populations lost 3 during migration out of Africa. The mites remember where we came from. In June 2022, research published in Molecular Biology and Evolution on Demodex folliculorum genome structure showed that these mites possess only 51.6 megabase pairs of DNA, second smallest genome among panarthropods. The gene loss is extreme. The mites are becoming obligate symbionts. They reproduce on us. They mate on us. They die on us. Vertical transmission from parent to infant ensures every generation inherits the mite colony. We do not choose this relationship. It chooses us. A 2024 study from the University of West Attica in Greece examined 184 individuals aged 18 to 97 using standardized skin surface biopsy. They measured mite density per square centimeter. 66% tested positive for demodicosis at above threshold levels. The median age was 51 years. Women represented 70% of subjects. The age distribution was statistically normal. Every decade of life showed positive cases. No demographic escaped. The same year, a meta-analysis covering January 2000 to January 2025 and published in Science Direct by Shahid Beheshti University, researchers found global Demodex prevalence at 35% with regional variation. Southeast Asia region showed 44%. Older adults above 60 hit 59%. Ophthalmology patients reached 57% because eyelid disease drives them to clinics where testing occurs. The mites concentrate where disease manifests. The percentage of males increased with patient age, suggesting different exposure patterns or immune responses between sexes. The researchers noted demodicosis presents with nonspecific symptoms that mimic other dermatological conditions, making diagnosis challenging. Burning, stinging, itching, redness. These symptoms could be rosacea, eczema, or mite overgrowth. Only microscopy or molecular testing confirms the cause. Most people never get tested. They live with symptoms or treat the wrong condition. The mites remain hidden in plain sight. Australian Aboriginal communities report prevalence up to 50% in some regions. Pacific Island nations struggle with endemic transmission. In developed countries, outbreaks cluster in institutions. A single crusted scabies case in a nursing home infected 70 contacts in one documented outbreak. The mites spread faster than containment measures. Mass treatment with single-dose ivermectin can halt outbreaks, according to studies published in Infection Control and Hospital Epidemiology in May 2018. But logistics and costs limit implementation. Globally, 65 to 130 million people suffer dust mite allergies. The prevalence continues rising in industrialized nations. In Germany, 1 in 4 people have dust mite allergy. In Hong Kong, 80% of asthmatic children are sensitized to dust mites. The allergens are ubiquitous. Dermatophagoids, Pteronosinus, thrives in humid climates. Dermatophagoids, Fahrenheit, dominates dry inland areas. Together, they colonize 84% of USA households. You cannot escape exposure unless you live in the desert. Even then, dust mites persist in mattresses and pillows shipped from humid regions. The allergens remain potent for months after mites die. Vacuuming stirs them into the air for 20 minutes. Opening windows after vacuuming helps clear allergens, but new mites colonize constantly. The war against dust mites cannot be won. It can only be fought to stand still. But diagnosis is difficult because early symptoms mimic many other fevers. The eschar, a black scab at the bite site, is the diagnostic clue. Not all patients develop eschars. Misdiagnosis leads to wrong treatment. The bacteria spread to lungs, brain, heart. Multi-organ failure follows. Survivors face long recovery periods. The mites are vectors but also reservoirs. They maintain the bacteria in wild rodent populations. Humans enter the cycle when they venture into scrub forest or grassland where infected mites wait on vegetation. Military operations in Southeast Asia during wars saw thousands of scrub typhus cases. The mites do not care about human conflicts. They feed when opportunity arises. Dermatophagoids mites in 84% of homes, causing allergies in 65 to 130 million people. Tromboculidae larvae biting millions annually and transmitting scrub typhus to 1 million. These are not theoretical risks. These are present realities. The mites are with you now, on your face, in your bed, on vegetation outside. Their populations fluctuate with seasons, with climate, with immune status. But they never disappear completely. Eradication is impossible because the mites reproduce faster than we can kill them, and reinfection sources are everywhere. Every human contact, every shared surface, every patch of grass. Anus, yes. The genome analysis revealed these mites accumulate waste throughout their lives, and only release it when they die. They have no way to excrete waste during their life cycle, so when they die on your face, all the accumulated feces and body contents rupture into your pores. That is what causes the inflammation spike in demodicosis. Not living mites feeding, dead mites exploding. The mites are becoming so specialized, so integrated with human skin ecology, that they are losing basic biological functions. They are simplifying. Genome reduction, gene loss, reduced repeat elements. They are converging on the minimum viable genome needed to feed, reproduce, and transmit. They are becoming extensions of human biology rather than separate organisms. The line between parasite and symbiont blurs. In another million years, Demodex may be indistinguishable from human organelles, or they may evolve into something else entirely. But right now, today, they are mites, and they are living on you. You cannot avoid chiggers without staying indoors every summer. You can reduce exposure, you can treat overgrowth, you can manage symptoms. But the mites will persist. They are resilient, they are ubiquitous, they are successful. They exploit a niche that humans created. Warm skin, sebaceous glands, shed keratinocytes, humid bedrooms, grassy parks. We built the habitat, they filled it. We are not victims, we are ecosystems. And like all ecosystems, we contain predators, parasites, and symbionts. The mites are all three depending on context. And if you shook hands with the wrong person, Scabies mites could be burrowing into your wrist. These creatures define the human experience as much as bacteria or viruses. We are not individuals, we are colonies. Every person is a walking habitat hosting thousands of species. The mites are just the ones you can see if you look hard enough, the ones science has named and studied. How many more species live on us undetected? How many undiscovered mites wait in follicles scientists have not sampled? The answer is unknowable, but one thing is certain. You are never alone. Even when you think you're clean, scrubbed, sterile, millions of tiny arthropods crawl across your skin. They are not leaving, they are home.